Hello, my name is Willette Streets, and this is Bio 225. Today's date is September 12th, 2018. Today I'm going to do the lab for aseptic technique and culturing microbes. After this experiment, I will have 3 broths labeled with 3 different specimens. One will be E. coli, the second one will be Staphylococcus epidermis, and the third one will be Saccharomyces cerevisiae. I don't know if I'm saying that right. Um, I have my supplies here. The first one I'm going to start with is the E. coli test. So I have my 3 nutrient broths labeled with 3 different experiments. I have my matches. I have my bleach water. I have my yeast. I have my Staphylococcus epidermis. Here I have the E. coli vial. And I have my alcohol in the pipette, which I use to sterilize in between. So the first thing that I'm going to start with is the E. coli. So the first thing I'm going to do is sterilize my alcohol. I have it here in the pipette. So I'm going to sterilize my pipette. And so while that is happening, I'm going to prepare the first specimen, which is going to be the E. coli. So this is my nutrient broth labeled E. coli, and I'm going to remove the cap from my nutrient broth. But first, before I do that, let me light my tea light. Okay, so we have this ready. So the first thing I'm going to do, I'm going to remove the cap from my nutrient broth, which I labeled E. coli. And then I'm going to pass the tip of the tube through the fire to sterilize it. Just like this. Hopefully you can see that. So I'm just sterilizing the tube by turning it. Okay. And so then I'm going to take my sterilized pipette and I'm going to get 0.25 milliliters of the broth. 0.25 here. Okay. So then I'm going to take my E. coli tube. I'm just opening it, taking it out of the little test tube. So this is my E. coli, and there's a tablet in here which is going to dissolve. So once I open it, I'm going to take the broth and squeeze inside. Okay. And then I will replace my pipette back in the alcohol to sterilize it. So once the solution is in here, I'm going to shake it really good until the tablet dissolves. Okay. I still hear something and it's almost dissolved. So I'm just going to keep shaking it. Once it dissolves, then I will transfer it back into the broth tube. Okay. All right, it's now dissolved. So I'm going to open it up, take my sterilized pipette, and then I'm going to get all the solution. Okay. I'm going to take all the solution. Let me put this here. And I'm going to place this back into the nutrient broth. Let me take the rest. There's still some left in this little test tube here. I want to make sure I get all of it. Okay. And I'll transfer that back. I'm going to take my pipette, place it back in the alcohol so I can sterilize it. Then I'm going to take my broth, which I placed the E. coli in, and I'm going to sterilize the tip again. So I take my tea light, and then I'm just going to sterilize the tip again. Okay. Then once I sterilize the tip again, I will close it back up. Okay. Close it back up. And then I'm going to place it back here. And so I also have 2 more to do. So the next one I will do will be the staphylococcus. And I'm going to repeat the same steps with the staphylococcus. And then the third one that I will do will be the Saccharomyces, which is the yeast. That will be the third one that I will do. And I will repeat the same steps as I did with the E. coli. And so once I'm done with that, repeating the steps again, being careful to sterilize in between, then I would incubate the 3 nutrient broths for 48 hours. And then I'm going to check for growth. If there's no growth within 48 hours, then I will add another 24 hours onto that to see if we have something. So I'm going to continue with my next 2. And my name is Willette Streets. This is Bio 225. Today's date is September 12th, 2018. Thank you.